Alright, so in this video, I'm going to be talking about translation. I'm not really going to go into detail about the difference between prokaryote and eukaryote translation because their process is basically the same except for the how, like the initiation and the release factors that are involved. But it's literally like generally the same. So let's do a quick refresher first. So translation happens in the cytoplasm for both prokaryotes and eukaryotes because transcription happens in the nucleus in eukaryotes first. That's where you produce the mRNA. And then the mRNA is going to go to the cytoplasm, and that's where translation happens. And the organelle that is responsible for doing the translation is the rough endoplasmic reticulum. Or you can just put it as rough ER. Now, why is it rough? Because the rough is just because of the ribosomes attached to the ER. If you guys have ever heard of the smooth endoplasmic reticulum, it's basically the same thing, except for the rough, there are ribosomes involved in it. And the ribosomes itself are the ones like doing the translation. So the ribosomes, we have something like a large subunit and a small subunit. Now, what's the difference? Basically, small subunit, I would say small or S. Small subunit is the one that decodes or reads the mRNA. While the large subunit, or L, or I might use L later, the large subunit is the one that catalyzes the formation of the polypeptide or the peptide bonds. So, this is basically what happens. But we also have one more thing. Wait. Let me go back to this. So, these ribosomes are mostly made of rRNA. Basically, RNA, ribonucleic acid. The R over here is ribosomal riboxynucleic acid. So, that's basically what it is. This ribosomal unit is mostly made up of rRNA. And then we have one more type of RNA that is involved, and that is the tRNA. The tRNA usually, like, if you see the slides, you'd see something like this. It looks like a clover leaf. Don't judge my drawing. It has the amino acid over here. It has the anticodons over here. The anticodons are complementary to the codons. So this is the tRNA. But for this video, I'm going to be drawing it like this. Like a letter T and something like that. Just to make it simple and not confusing. And then, you've also heard of the A site, P site, and E site. The A, the P, and the E. It's an imaginary site so that it's better to understand how the movement of the RNA and what site is the peptide bonds gonna be forming. It's in the ribosomes. It's like the imaginary sites in the ribosomes. You can think of it like this is the A site, this is the P site, and the E site. Now, what's the main difference between the three? A site basically accepts new tRNA with the new amino acid. And then P is processing or like holding the tRNA with the amino acid until a new amino acid comes in. So you can think of it, A accepts, P processing, and then E is where the exit site. That's where like the tRNA is gonna be ejected. So exit. So I don't know if you have other ways to remember it, but this is my way. I know the A site accepts new tRNA. P site processes the tRNA, and then E site is the one that is the exit site. So let's visualize this. I prepared something over here. Alright. So, first of all, we have the untranslated region in the 5' and the untranslated region in the 3'. Now, in the untranslated 5', we can find the Shine-Dalgarno sequence, that's for prokaryotes, and we can also find the 5' cap in eukaryotes. So basically, those are untranslated region because those are not part of the during translation, those are not going to be translated. Those are just sequences for maybe for stabilizing the RNA itself. So let's do, first of all, the small subunit. I'm going to draw it like this. Or this is a small subunit, right? The small subunit already has the tRNA or the initiator tRNA, which is usually methionine. So it already has the initiator tRNA. Now the initiator tRNA is already on the P site of the small subunit. The small subunit is going to keep reading through the untranslated region, depending on what kind of cell it is, maybe prokaryote or eukaryote, until it's going to reach the codon that codes for the methionine. So this is the codon, AUG. So once it reaches here, it's going to bind there. It's going to bind to the AUG. Well, not necessarily bind, but more like, it's like the anticodon is going to recognize the codon over here, because anticodon and codons are complementary to each other. So this is what happens next. So remember earlier I said there's a A site, a P site, and E site. So this is the E site, and this is the P site, this is the A site. So on the P site, as I said earlier, the initiator tRNA is already on the P site of the small subunit. Once it finds it, it's gonna bind it here, and then the large subunit is also gonna bind to the small subunit to make a complex to start the catalytic portion. So now, what's next? We have the A site. What's on the A site? It's GAG. If you look at your codon chart, I believe it's glutamic acid, which is E. So glutamic acid is gonna come in and then stay here, right? And then within the A site also, that's where the new peptide bond forms from the methionine to the glutamic acid, or E, or whatever amino acid it is. So it's gonna form a peptide bond, and then this tRNA and the amino acid itself is gonna dissociate within these first few steps. If you haven't watched the quick overview of peptide bond formation, or if you're not familiar with it, I would suggest watching it so that you kind of understand how the peptide bond forms. Alright, let's get back to this. So let's say, so the E, or the new amino acid, enters the A site, and then the peptide bond is going to form between the amino acid on the P site to the A site. So now, after that, small subunit is going to translocate, and the large subunit is also going to translocate. It's going to move from 5' to 3'. So this is what it looks like now. If you look above here, this is the A site, the P site. Now, the A site and the P site has moved on this side. A, P, and E. So now what happened to the tRNA of the methionine? So the tRNA of methionine is going to be ejected at the E site. So V, yeah, like that. And then you see this, the glutamic acid from the A site moved to the P site now. So what's next? On the A site, we have the ACU, which I believe is threonine. Just if I'm wrong, just correct me correct it with your codon chart. So threonine is gonna come in and then go to the A site. Same thing. this amino acid on the P site is gonna make a peptide bond with the one in the A site. And then this amino acid in the P site also is gonna dissociate with this tRNA over here. And then just continue the process. Now look at this, on the P site we have the threonine tRNA. And then what's next? We have CAU, which is, I believe, histidine. So histidine is gonna come here to the A site, which will be here. Same thing, a peptide bond will be formed between the threonine and the histidine. And then dissociation will happen between the tRNA of the threonine and the threonine itself. So after that, what's next? We have UAG. UAG is the stop codon. So this is something that's different. We have a protein called the release factors. Release factors look like a tRNA as well. They kind of mimic the tRNA so that they can, it's like, I don't know how cells do it, but yeah. So release factor is a protein. It's not a tRNA. Now this protein is gonna bind to the stop codon. So in this one, I would just say this is RF. You know what, let's put it different color. RF, the release factor is gonna go here to the A site. And once it goes to the A site, it's gonna kind of like give a signal. It's gonna give a signal to the whole ribosomal complex to say, oh, you're done. Don't stop catalyzing the peptide bond formation anymore. You don't need to translate anymore. So basically, it's gonna release everything. That's why it's a release factor, because it's a protein that releases everything. It's kind of like it stimulates everything to release. So what happens is that your large subunit is gonna dissociate. Your small subunit is gonna dissociate as well. And then your polypeptide chain is also gonna dissociate. So that means you have a chain of polypeptide. So yeah. So this is basically the final product of translation. Now this is not yet a protein. Maybe it's a protein already. It depends. All proteins are polypeptides, but not all polypeptides are protein. So we would call this a polypeptide. And then depending on modifications after translation, it can become a protein. So yeah, we have the N-terminal, our amino acids, a C-terminal, and then we also have the peptide bonds between each amino acid. So yeah, that's basically translation. It's similar for both prokaryotes and eukaryotes. They might have some specific stuff, like the ribosomal units has specific structure, and then there's also like specific sequences of the untranslated region, like the Shine-Dalgarno sequence or the 5' capping. But yeah, this is basically translation. I'm gonna try to summarize it again. Let's just do it here. Small subunit with the initiator tRNA is gonna read through the untranslated region, and then it's gonna go through until it finds the codon that translates for the methionine. So when it translates, the large subunit is gonna come in to make a complex with the small subunit, and then new tRNAs is gonna come and then enter through the A site. It's gonna get processed on the P site, and then it's gonna exit at the E site. And then it's gonna keep going until it reaches a stop codon. Now on the stop codon, tRNA will not bind to it except for a release factor. The release factor is gonna bind to the, it's gonna go to the stop codon, and then it's gonna signal everything to stop. You're done translating, and then your large subunit, your small subunit, they're all gonna dissociate with the mRNA. Your polypeptide is gonna dissociate with the subunits, and then like this large subunit, this small subunit, this mRNA, they might get degraded, they might get reused again later on. So yeah, that's basically translation. And I really hope it helped you. If you're confused, feel free to email me, email Dr. Zolman. And as for memorizing the amino acids, I would suggest watching Ken's video. She has a good way to memorize the amino acids. So yeah, thank you for watching.